Okay, thank you. Good afternoon, everyone. I'm going to present the preliminary results of the second chapter of my PhD research project. And as Ivan told you, this presentation is named Diversity and Evolution of a New Genus of Tiger of Tarantulas from Southern Mexico and Central America. So, the tribe of Peloponnese is currently composed by 15 genera and almost 100 species. And one of the representatives of this tribe is the genus Davus, and the tarantulas of this genus are characterized by having a dorsal coloration pattern of the abdomen composed by transversal stripes, orange and black, which gives them the common name of tiger of tarantulas. And the last revision of the genus was conducted in 2016, and after this revision, the genus is composed by 4 species. Davus pentaloris from southern Mexico to Nicaragua, Davus rufisa from Nicaragua and Costa Rica, Davus fasciatus from Costa Rica and Panama, and Davus santos only known from one locality in Panama. And the last taxonomic revision was only based in morphology, and especially those that of the dorsal coloration pattern of the body, but also in the sexual structures of males and females. So after this revision, Davus pentalori had a widespread geographical distribution, and also it present a high morphological variation throughout this distribution. However, recent studies in megalomorphs has shown that these spiders usually have a conservative morphology and also a limited dispersion capability, which is usually caused by their sanitary habits. And this limited dispersion capability usually produce localized endemism and high genetic divergence. So this evidence raises a question if there is underestimated diversity in this group of tarantulas. So I started revising the material deposited in the National Collection of Araneae at the Institute of Biology at UNAM, and I found many specimens collected in different parts of Mexico, and I found also a very high morphological variation in the sexual structures of males, especially in the male papal bulbs and the tibial apophysis. And because these structures are very important taxonomically to separate the species, all this variation raises the question if there is only one species in this group of tarantulas. So the research question of the first chapter was that do all the population of Davus pentaloris that have been reported from southern Mexico and Central America belong to a single widely distributed and with considerable morphological variation species, or if this morphological variation is showing us that there are more than one species in this group of tarantulas. So to try to solve this question, I revised many specimens deposited in national and international collections, and also I conduct fieldwork in many localities of the states of Guerrero, Oaxaca, Veracruz, and Chiapas, and also I could include 2 samples of the populations of Central America, one from Guatemala and one from El Salvador. I conduct extremely morphological revision. Also, I took many photographies, and also I took quantitative data to see if these data support the hypothesis that there are more than one species in this group of tarantulas. So because we wanted to include molecular data, we include a partial segment of COI of 963 base pair, and all the molecular protocols were conducted in the National Laboratory of Biodiversity at UNAM. For the species elimination, we applied the general lineage concept of a species, and we used 5 species elimination methods based on morphological and molecular evidence that were used as secondary criteria. And the species delimitation was based on the congruence of the different results with the different evidence. So we had the morphology, and with the molecular data, we used 2 methods based on genetic distances, ABGD and neighbor joining, and 2 methods based on freeze and qualescent theory, GNYC and BPTP. And we compared the different results to arrive at an iterative solution to propose the species hypothesis. So the morphology in the males was very informative. We have found very marked differences in the male papal bulbs and the tibial apophysis. And considering the available data, we identified 13 morphological different morphospecies. And also we found somatic characters in the dorsal coloration pattern of the body and other structures that support these morphospecies. So by comparing the results obtained with the molecular data and the morphology, we determined that in the complex Davos pentalaris, there are 13 confirmed candidate species and 3 unconfirmed candidate species. Because of 3 populations, we could not include morphological data because we only had juveniles and females. And we found that the females had very variable structures, so sexual structures, so they are not reliable to propose species limits in this group of tarantulas. However, what happens with the rest of the species of the genus Davus? So we reconstruct a gene tree of koi, and our expectation was that all the species of the genus Davus would be recovered as closely related species. However, this was not the result, and because we only had one gene, and we are observing here the history of koi, we need to see what's happening here by incorporating additional molecular markers. And also we wanted to see what's telling on the morphology. And we found very marked differences in the sexual structures of males, not only in the general morphology of the male palpal bulb and the tibial apophysis, but also in the number and morphology of the keels of each male palpal bulb, and also in the shape of the branches of the tibial apophysis. So this is the tibial apophysis of Davus fasciatus, the type species of the genus, and this is the morphology of the tibial apophysis of the complex of Davus centauralis. And also other marked differences is that the species Davus ruficeps doesn't have these structures. So all this evidence raises the question: is the genus Davus as currently composed monophyletic? So we try to obtain 5 molecular markers, and also to include other members of Apalopini to try to solve the relationships within Davus and also with other members of Apalopini. And we include some representatives that also had a dorsal coloration pattern of the body. So this part of the project was very affected by the pandemic situation. At this moment, we only were able to obtain 3 of the 5 molecular markers. However, the preliminary results show that all the species that belong to the complex Davus pentalaris belong to a new genus of the tribe Apalopini. And also the species Davus ruficeps and Davus fasciatus are not completely closely related. So, however, we need to incorporate the other 2 markers to see if the branches in the mid part of the tree have a higher support, because at this moment some of the branches have a low support. And at this moment, I'm working with the taxonomic revision of the genus by describing the new species that were delimited in the species limitation, and also to see if some morphological characters are very useful to incorporate them in the phylogenetic analysis to have a total evidence analysis. So our preliminary conclusions are that the morphological and molecular evidences seem to indicate that the genus Davus is not monophyletic, and also it is necessary to incorporate the rest of the molecular markers to the analysis to try to solve adequately these relationships. And the preliminary results indicate that the 13 confirmed candidate species that were delimited in the species limitation methods belong to a new genus of Daguerreotyph tarantulas. Also, it is necessary to include the morphological data in analysis to see which of those characters are phylogenetically or have a phylogenetical signal, and also which of these characters are important to diagnose the different genera that result from this revision. Also, it is necessary to complete the taxonomic revision of the genus and the other new genera and species as a result not only from the phylogenetic analysis, but also to the species delimitation analysis, because a week ago I was able to conduct some field work in Chiapas, and from those localities I could not include in the species delimitation chapter. So I'm going to try to introduce those specimens in the next analysis. So I want to thank many people that have contributed to this project, especially my lab mates, Dariana Guerrero and Daniela Ble. Also all the members of Viernes de Arañas. And also I want to thank CONACYT and the American Araneological Society and the International Society of Arachnology for the scholarship given to me to finish this project. So thank you.
Thank you very much, Daniela, for this interesting talk. And now, if people have questions, they can type into the chat. You're getting a lot of compliments. I'm sure people are clapping for the hums. And while no one has asked a question, I would like to ask one. So you still have to incorporate morphological data into the analysis, but the characters that supported Davus as a single genus, what do you think is going to happen when you put them into the analysis? Do you think, because you show Davos is not monophyletic, could they be convergences, or there are actually no characters that supported Davos before?
I think that there were not enough characters to support the monophyly of Davos before, because the main character that was used to put all the species together was the presence of a dorsal coloration pattern of the abdomen. However, in the different species, the dorsal coloration pattern of the abdomen is very different. So I think also that these characters in the dorsal coloration pattern could be useful to diagnose the different genera within this currently known as Davos.
Okay, thank you. Darko Kotoras is asking what is or what are the synapomorphies of the new proposed genus?
Actually, the genus Davos was not previously incorporated to a phylogenetic analysis. So all the diagnostic characters that were proposed to diagnose the genus were not proved in a phylogenetic analysis. So the general morphology of the male pulpal bulb, some keels, however, they were not extremely revised, or I don't know how to tell it, because for example, some keels in Davus fasciatus have tiny dentitions, and those dentitions were not incorporated in the original descriptions. So all these characters are new, and I'm going to see if these characters are have a phylogenetical signals to see if there are some of them are synapomorphies of the genus.